Hello and welcome to Nikolay's genetics lessons, and today I'm going to talk about how to understand gel electrophoresis results. And here's a problem. A segment of DNA has two restriction sites, 1 and 2. When incubated with restriction enzymes 1 and 2, 3 fragments will be formed, A, B and C. Which of the following gels produced by gel electrophoresis would represent the separation and identity of these fragments? And in order to solve this problem, basically everything you have to know that DNA, and here is a double-stranded DNA, has slightly negative charge. You see negative charge here, 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 and here. And also on the corresponding strand of the DNA, we also have negative charge per every base. So, because on the overall double-stranded DNA have negative charge, that means that when we run a gel, our DNA would be loaded on the negative side where negative charge would be applied, and our DNA would move in the direction of the positive charge. As you remember, the same charges would repel each other and different charges would be attracted. So we load our samples of the DNA in these wells here, and negatively charged DNA would travel in the direction of the positive charge. Now, if you would take a look at this double-stranded DNA, you would see that when two restriction enzymes would cut this fragment of the DNA, we are going to get DNA fragments of three different sizes. Fragment C would be biggest and heaviest, and that means that this fragment would travel the shortest distance. Just like imagine three people with different weight. One would be overweight, another would be medium weight, and the third one would be lightweight. If they would run, after 1 minute, we would expect that the guy who would be overweight would travel shortest distance, and the guy, for example, that is going to be lightest would also travel the greatest distance. So let's now take a look at these gels. So we expect that fragment C would travel shortest distance from the well. What we see, for example, in this gel, we see that fragment A traveled the shortest distance from the well, and that means that this is not the correct answer. Here we see that C traveled the shortest distance, and this is correct. Here we see B traveled shortest distance, and this is not correct answer. But here also seems like correct answer and wrong answer here. So let's now analyze this gel and this gel. So second heaviest would be fragment A. So we see fragment C here and fragment C here. Once again, heaviest fragment traveled shortest distance. Second heaviest is on the second place, and the lightest traveled longest distance. And this is would be the correct answer. And if we compare now with another variant that is here, we would see that the order of the band is wrong. Heaviest, then have to be fragment A. Instead we have the smallest fragment here in the second place. So this is wrong answer. And the rest we excluded at the first step. And this is all for today. Thank you for attention. Please subscribe for my new videos that I post almost every day. Thumbs up if you like this video. Please write your comments, questions if you have any. Share this video with your classmates, and see you in the next video. Goodbye.